Okay. I hope everyone can hear me. If you can hear me, just please put something in the chat. Okay, I hope everyone can hear me. Hello and welcome to this live session. I'm realizing very quickly that my network might not be so great. So I hope that I'm coming through very clearly. Awesome. So I can see that many of you can hear me. And I don't know if you can see my video, but I hope that you can. So Dr. Demi, if you have been viewing my videos on YouTube, this is what I look like. For those of you who don't know me, most of my students where I used to teach obviously know what I look like, but I don't see any of them on this video. So I'll just go ahead and introduce myself. I am Dr. Demi, not the kind of doctor that can prescribe medicine, but I have a PhD in engineering. So that's where my doctor comes from. And I have taught biology for some time. I see some of you are still struggling with being able to see me, so I'm not really sure what is going on. Okay, I'm just going to try. I think this might be a YouTube problem. Let me see. This might be a YouTube problem. Let me see. Okay, I'm not sure if this is better for those of you who are. If you can't see me and you can hear me, maybe that would work. I think let me just try something first, the most important thing, which is what we have to do today, and that is to share the paper I would like for us to work on together. So let me just see what that would be. So I see that now you can see me. Awesome, great. All right. I'll just reintroduce myself and then we will work out how to share the screen later. So I'm Dr. Demi. Like I said earlier, I'm not the kind of doctor that can prescribe medicine. So if you have any kind of ailment, please don't ask me because I can't. I have a PhD in chemical engineering and I worked as a biology teacher at A levels for 3 years at an academy here in South Africa. So I live in South Africa at the moment. And I just really enjoy being able to make learning accessible to everyone. And that is why I decided to do this live. So I hope that you find it useful. I figured for those of you who are writing your exams in October, November, this could be a great opportunity for us to just look at some past questions together and see how you would approach those questions. So I chose a paper because this is our first live and I obviously don't know what kind of crowd I'll be getting. So I've chosen a multiple choice paper. And since you guys are very active, I can see that in the chat. What I'll do is I'll share this paper and every time we look at a question, just post what you think the answer is. And we will then offer an explanation for why that is possibly the answer. So I hope that is helpful. Claire, if it's clear, please just say so in the chat. And I see Philo Maged, you're welcome. I'm glad that you're using the videos to prepare for your exams. All right, let's see. I'm just going to look for the paper that I want to share. So let's see if this opens up. Okay. So can everyone see the screen currently on? Can you see the web page? Not the web. Can everyone see the multiple choice paper that I have just opened? Or are you seeing something else completely? Okay, so I see here that it's showing my Windows Explorer, but I'm hoping to share with you the multiple choice paper. So if you can't see the multiple choice paper, please just let me know. Okay, so you all see this paper, May, June 2019, that is open here right now. And everyone can see question 1, I hope. Can we all see question 1? Just going to give a few seconds to... Oh, so you can't see the PDF file. All right. What's going on there? I'm just going to try that again. And it's actually quite a mission with... Okay, I'm going to try to share it again. Is this better? I voting, but can everyone see this now? I hope that's a yes. Okay, I'm guessing that's a yes from everyone. Great. Okay. Okay. So this is a 2019 May June paper, I believe. Yes, this is a May June 2019 paper. And what we're going to do is just look at some of the questions. Well, I'll try to go through the whole paper on this live, but I don't want to keep you guys for too long. So I'm hoping that at most we will spend 90 minutes together. But I hope that by the time we are done, you've picked up some tricks on how to answer multiple choice questions. Now, this is very important because, so I say someone says to zipper, so I'm just going to try to make that bigger. So again, just want to say that some of the papers that you will have to do when you go into your exam, some of the papers that are actually really difficult, rather, are the paper 1 questions and also the paper 4 questions. So many students struggle a lot with paper 1 and paper 4. Most of the success that students have in CIE is usually in paper 2, paper 3, especially if you've been practicing your lab experiments, and in paper 5 because it's an analysis paper. And I know for some of you, paper 5 is difficult, but we'll get to that in some of these lessons. But for now, I really want to improve your ability to take a paper 1 paper. And that's because paper 1 is confusing for many students. So what I'm going to do here, the reason why I've chosen this, I'm going to be using my pen here. So I'm scribbling on the page now, and I'm just going to give it a few seconds. And I want you to tell me if you can see this jargon I've drawn here before we proceed, so that way when I start to write, you're able to see. So if you see the annotation, that's the blue pen I've just drawn. Please indicate in the chat that you can see it. Perfect. Okay, good. So we are ready to go, and I'm very excited. If you ever encounter any of my past students, they'll tell you I love questions a lot. I love past questions. So this is very exciting for me, and I hope that you also enjoy it because it makes it easier for you to enjoy the paper. So I'm just going to zoom in a little for those who might still not be able to see properly. And so let us get into it. Question 1. A student was told that the actual length of a cell structure is 5 micrometers. The student was asked to state an equation that can be used to calculate the magnification of an electron micrograph of this cell structure. The student used some of the letters Q to U in the equation. And here we have definitions of Q to U. Q is the length of the cell structure image on the micrograph in centimeters. R is the length of the structure in millimeters. S is 1,000. T is 5. U is 5. Okay. What is the correct equation to calculate magnification? Now, when students see questions like this, many times, many students get scared because it's not the usual question that they expect. And this is why I love these kinds of questions, because they make you think about what you know. Now, I want you guys in the chat, first of all, to type in what the equation for magnification is. The paper is May, June 2019, paper 1. What is the equation for magnification? Just the general equation, not based on this question. Just post it in the chat what you think magnification, or what you remember the formula for magnification is. I'll give you a few seconds. Good. So I see there, image size over actual size. That is correct. So you know you're dealing with image size over actual size. Now let's look at the question in a bit more detail. The student is told that the actual length of a cell structure is 5 micrometers. When you are calculating magnification from a micrograph, by the way, for those who might not be sure, a micrograph is when they give you a picture of the image, okay? So it's like an electron micrograph that you can look at. The length of that image is going to be the image size, okay? And the actual length, which is already given to you here, is 5 micrometers. I hope everyone is following this. Now, if you look at the symbols that have been used here to represent the different things that you would do, U is equal to 5. Okay, so you already know in this case then that magnification is equal to I divided by 5. But the question here is, what is I? Now they say here that Q is the length of the structure of the cell structure image in centimeters. And that's not a bad thing. But look at what they've also given you. They've also given you R, which is the length in millimeters, and they've given you this value of 1,000. All right. Now, if you go from centimeters, you would first have to multiply your centimeters to take it to meters, and then from meters to millimeters, from millimeters to micrometers. So you don't want to go through all that, and they also haven't given you the option to do that. What they've given you, however, is how to go from millimeters to micrometers. If you want to go from millimeters to micrometers, what do you do? Just put it in the chat. From millimeters to micrometers, what do you do? I'm waiting. If you're going from millimeters to micrometers, what should you do? Multiply by 1,000. Well done. Good. So we know then that if we have R, which is the length of the cell structure in millimeters, and by the way, this is the image length, because remember, when you're looking at the picture, you're looking at an image, all right? So it's not the actual length. The actual length has been given to you. So the length of the structure in millimeters, and you also have 1,000. So what does that tell us about what I would be? What is I in this case? Using the symbols now. I'll just wait a bit because I know the network is not too great. So I'm just going to wait a bit. And I want you to tell me what I would be based on the symbols that we have been given. We've agreed that if we go from millimeters to micrometers, we multiply by 1,000. We've been given the length in millimeters. Good. R times S. So that means that our image size is R times S. I hope everyone is following. If you're not following, please don't be shy. Put it in the chat and say, please explain again, and I'm willing to do that. Don't be ashamed. I always tell my students, there are no stupid questions. So whatever question you have, even if you feel like it's something that you should know, please, by all means, post it there so that I can clarify whatever it is that you don't understand. So we know that I is equal to R over S. And we also know that our actual size, because let's just write the formula here, M is equal to I over A. Okay, so we know that I is R times S, and we know that A is 5, which we've been given here, and that is U. But look at this now, we have a problem. When we look at the options, there is no R times S divided by U. Can everybody see that? So obviously, this doesn't mean that we're wrong. It means we need to think of a different way of writing U. And the different way of writing U is to look at what else we've been given, which is T. T here says equal to 1 over 5. So if we were to redo this, let's say, for example, we did it this way. So magnification is, I'm just erasing here. So I'm just going to say here, magnification is equal to I over A. Okay. We know that I is equal to R times S. And we know that A is 5, which is equal to U. But another way of writing this, instead of writing divided by 5, is that we could multiply what is up here by 1 and divide that by 5. Because when you multiply this by 1, all right, R times S will still retain its value, and that way you're able to divide by 5. And in that case, your answer then becomes D, which is R times S times T. Did everybody get that? If you got that, just put in the chat like, yay, you did. Come on now. And if you have questions, also post them there. So I'm guessing some network challenges, so the chats are not coming in as timely as we would love for them to, but I really hope that everyone understood that. I'm now going to go to the second question, but again, if you have any questions about this, we can come back to it, all right? But the answer for number 1 here is D. Okay, so everyone says they got it. Well, those of you who responded. Now let's look at question 2. Now, these kinds of questions are usually very common in CIE, where you have to deal with like a feature and an organelle or something, all right? So I always say to students, don't be confused, and let's just look at the question and I'll see what tips we can apply here. So it says, which features of cilia and root hairs are correct? All right, so which features? Do you guys know what the cilia are? I hope you know what the cilia are. So the cilia are like thin finger-like projections that you can find sometimes in the gas exchange system. You can find them in different parts of the body on the surfaces of cells, and they help in reabsorption. They help in increasing the surface area. So I've just given you one of the answers. The root hairs also help in increasing surface area, and they are found in the plants. So when the roots of a plant go into the soil, you find these very thin hairs that help with the absorption of water. And if you've done transport in plants, this would be very, very easy for you to remember that water moves through the root hairs, goes through the endodermis, reaches the Casparian strip, and then enters the xylem. But we will not go into all that detail just yet. But let's see what features of cilia and root hairs. So that they increase surface area. Yes, they do. They do increase surface area. They can't be resolved with the light microscope. They contain vacuoles, and there is more than one present on a cell. So now, in this case, when you're dealing with this, this is one of those questions where Students get really confused very quickly because cilia and root hairs tend to be very similar in function, all right? But if you look at this, contain vacuoles. The cilia cannot contain vacuoles. I'm going to use a red pen for the ones that we don't agree with. So the cilia here, that can't contain vacuoles. Why is that? Because the cilia are not exactly cells. They are projections that are on the surface of cells, so they can't contain vacuoles. So that tells you that option B cannot be your answer, as well as option D. Okay? I hope that's clear. And the reason I always use this method is I try to limit for students the amount of things you need to pay attention to when you're trying to choose the right answer. If you wanted to go through the route of saying, okay, I want to look at each and everything and not eliminate anything, you might get confused very quickly. But what we've done now is that we have opened things up, or basically eliminated things, so that now we're only considering two options. I call it the 50-50 split. You kick out the options that are not possible, and you look at only the options that are possible. Okay, so now let's look at option A, because we know option A might be possible based on just this trait or this characteristic. Cilia increase surface area. Well, they do that. They cannot be resolved with the light microscope. That's true. Contain vacuoles, there's root hairs, more than one present on a cell. Now, root hairs are not present on cells. They're present on the whole root itself. So the root itself has many cells within it. So that makes this answer an impossibility. And you're left with only C. And that's the correct answer. Did that look like magic? I hope not. I hope that was a bit clear on how you do the elimination. Okay, it's a bit silent in the chat, but I hope that you got that. Question 3 again, I don't want to keep everyone for too long. I want us to finish this as quickly as we can. Let's look at question 3. What are the functions of microtubules? So here I'm actually just going to leave you guys to tell me which answer you think it is, and I will explain your answer for you or tell you why your answer might be wrong. So which are functions of microtubules? Is it A, which is 1, 2, and 3, allowing movement of cilia in the bronchus, attachment of centromeres during metaphase, moving secretory vesicles around the cell, or is it B, which is 1 and 2 only? Which one do you think the answer is here? Just going to look in the chat and see what you guys come up with. And please don't be shy. Don't worry about, getting the wrong answer. The point of this class is to help you. So you can't be helped if you're too worried about what anyone in the chat might think. So just post your answer, give it some thought and post your answer, and I'll tell you if you're right or wrong. Okay. You see Fatima's posted an answer. The rest of you show up, maybe. Oh, it's a bit too quiet in here. All right. Okay. So the correct answer here is actually A. And these are obvious. Okay. They allow centromeres to attach during metaphase, because you remember that the microtubules aid the formation of the spindle fibers, which then allow for chromatids to be pulled apart. They also move the secretory vesicles around the cell. But something else that they do, which is not commonly known, is that they allow for the cilia to move in the bronchus. And so that makes the answer here A. And I know for many of you, you're like, wait, what? But these are some of the things that you discover when you do past questions. And something else that I think I should just say as a side note, now that we're here, is that if you want to excel in your exam, all right, if you want to really, really excel in your exam, the best thing to do is to practice as many past papers as possible. Your success does not lie in how often you read your textbook or what information you can recall. It actually lies in being able to determine what kinds of questions you should expect and the extra information that's not in your textbook that comes out when you do questions. So for many of my students, I advise them, watch the YouTube videos. After watching a video on a particular chapter, get questions on just that chapter, try them out and see how you do. What that will help you do is to identify the things that you know and the things that you need to work on and the things that are not even included in your textbook, but turns out that you need to know, such as this one. We would have thought that microtubules help with the movement of cilia, right? Let's look at the next question. Okay, I love this particular question, and I see that CIE brings it out a lot. So I want you guys to tell me, here it says some cell structures are listed. What is the correct order of these cell structures when listed from the smallest to the from the largest, to the smallest? So have a look at these. You have the ribosomes, the chloroplast, the nucleus, the mitochondria. What do you think the correct order is? From the largest, so the biggest one, to the smallest one. And I think we can start that first of all by saying, well, which organelle is the smallest? Maybe let's start there. You can just type the name. Which organelle do you think is the smallest? Okay, I see many of you already picking B. All right. Anybody else? I'm going to explain the answer. Yes, indeed, B is correct. But let's look at why B is correct. And this is the point of these classes, okay? Ribosomes are the smallest if you look at this list, okay? So it means that 4 has to be the last number. That, again, if you use the method of elimination, removes C and D as possibilities. Do you see what I've done there? So now you're left with looking at just A and B. Okay? Always use this method because it makes you more likely to choose the correct answer. So if you want to think of it mathematically, instead of having a 25% chance of choosing the right answer, you now have a 50% chance. And that is a very good chance to have. So now let's look at the sizes of the organelles. The nucleus contains the DNA, it's the powerhouse, blah, blah, blah. It's actually the largest organelle inside the cell. That is followed by the chloroplasts. They have the grana, the thylakoids, and all of these other things. That makes them second largest, followed by the mitochondria and then the ribosomes. A might be the answer for many people, but please don't pick A because the mitochondria is not bigger than the nucleus. The nucleus is actually the largest because it contains all of that information. And I know that might be confusing because when you look at it in a cell, it looks small. It's like this little circle, but that's because you're looking at it in an image. The nucleus actually is a big organelle, and it's followed by the chloroplasts, the mitochondria, and then the ribosomes. So the correct answer is B. Well done, everybody. Let's look at number 5. We still have a bit of a way to go, but I hope that we are, I hope these are helpful for you guys. So please stay engaged. Don't disconnect. Don't be like, oh my God, this is too hard. All right. Let's look at this one, number 5. Which cell structures have ribosomal RNA? I want you guys to tell me. Chloroplast, mitochondria, nucleus, and the rough endoplasmic reticulum. So for an organelle to have ribosomal RNA, what should it have? Okay, I was about to say, oh, I see the right answers, but I see the first one. For an organelle to have ribosomal RNA, it has ribosomes. Okay, that's a quick way to think about it. So instead of saying, what does ribosomal RNA do? Just think of the fact that it says ribosomal RNA. So definitely it has ribosomes in it. All right. So in that case, which of these organelles have ribosomes? Think about it very carefully. You can change your answer if you want to. Which of these organelles have ribosomes, either inside of them or on them? And that would guide you to the right answer. I'll give you a few seconds. I know the network might be lagging a bit, but think about it. Which of these ones have ribosomes? And what would your answer then be if you thought about it that way? Everyone else seems shy because I only see Fatima's answer. But I'll go ahead and tell you the answer is A. Chloroplasts have ribosomes because remember, if you think back to the endosymbiont theory, and I hope you all know this theory, the endosymbiont theory basically suggests that chloroplasts and mitochondria used to exist on their own as prokaryotes. All right. And because they existed as prokaryotes, they basically made all of their own things. So they have 70S ribosomes, if you remember. And obviously, for them to be able to make those ribosomes, they would have ribosomal RNA. So chloroplast and mitochondria actually have ribosomal RNA. The nucleus also has ribosomes in it. All right. The nucleus has ribosomes in it. And the rough endoplasmic reticulum, if you remember it, looks something like that. And it has ribosomes sitting all over it, which is the reason why it is called the rough endoplasmic reticulum. So in this case, the answer would then be A because they all have ribosomal RNA. They all have ribosomes, so they would all have ribosomal RNA. I hope this explains the question better. All right. And I know, students, the nucleus is a confusing one because you're usually not told explicitly in class that the nucleus has ribosomes in it, but it actually does. So yeah, let's look at the next question. So let's look at this one. A cell structure in the macrophage destroys bacteria. Some bacteria stop this cell structure from functioning. Which cell structure is the macrophage is stopped from functioning by the bacteria? The question here is which cell structure, the real question is which cell structure destroys bacteria? If you look at any one of these, I think this is an easy one. I don't want to be preemptive, but I hope that you all get this one right. What is the answer to number 6? Come on now. I'm waiting. What's the answer to number 6? Which cell structure destroys bacteria? Or if you want to think of it in an easier way, which one digests bacteria? Yes, you are all correct. The answer is B, the lysosome. The lysosome is known for digesting bacteria. And if you think of it if you think of what I did when I asked you the question again, is I went back to the beginning of the question. A cell structure destroys bacteria. Okay? Which cell structure is this? That's actually what they're trying to ask you, even though they go around in a what's it called? Should I call it the scenic route to get you to what the question really is? They are asking you for the cell structure that destroys bacteria, and that can only be the lysosome. Just for the sake of making things clear, the Golgi body is responsible for the modification of proteins. The ribosome is responsible for protein synthesis. It's the site of protein synthesis. The vesicles are basically transport agents that are used to carry proteins outside of the cell once they have been made. So there's no way any of those would be involved in the digestion or the destruction of bacteria. So that makes the answer over there B. All right, let's look at number 7. Oh, I love chapter 2. So again, another thing I want to draw your attention to, everybody, is that when you look at your CIE past questions, you will notice that the questions follow a sequential order. So from chapter 1, you move on to chapter 2, you move on to chapter 3. So that might help you when you're thinking in the exam to say, what question or what chapter am I on? And that can help you when you determine what your answer should be. Because sometimes the questions overlap or the chapters overlap in content, but when it's chapter specific, you're able to limit what you should be looking at. That's just a tip for you. I hope that you find it helpful, but it's a tip that you will really find useful when you do paper 2 versus paper 4. So just putting that out there. Let's look at number 7 and let's see if you can tell me what the right answer is. So it says a solution of amylase was added to a suspension of starch. What does amylase do, everybody? Just put it in the chat if you know what amylase does. I'll give you a few seconds to do that. What does amylase do? Waiting. I know this is a network issue, not that you guys are not engaged, but I'm hoping that you know what amylase does. I don't want to just give you all the answers. Breaks down starch. Thank you so much. Yes, that's correct. Amylase is an enzyme that breaks down starch. All right. So if amylase is breaking down starch, starch is made up of amylose and amylopectin. Amylose and amylopectin are made up of repeating units of alpha glucose. So basically, the end point of all of this would be glucose. So it says here, after 30 seconds, 3 samples of the mixture were tested with iodine solution, Benedict solution, or with biuret reagent. I hate saying that word. I feel like it's quite annoying. And then it says, what are the expected results? All right. So here in this case, you have starch. What does this reagent test for, the biuret reagent? What does it test for? Oh, I'm just seeing the answer, the amylase or amylose question. But I want you now to tell me, what does biuret reagent test for? Yes, it tests for proteins. That's good. So this reagent is used to check if proteins are present in a sample. And if proteins are present, what is the positive color that you would see? What's the positive color for a protein test? The positive color for a protein test is purple. All right, good. Well done, everyone. So let's then go back to our question and see what the answer should be. All right. So if we break down starch with amylase, we know for sure that we are going, and we tested with iodine. Okay. So it says amylase was added to a suspension of starch. After 30 seconds, the mixtures were then tested. So obviously, if we add amylase to starch for just 30 seconds, there's a possibility that we will not have the complete digestion of starch. So some starch would still be present in the sample. That then tells us that, and you know when starch is present and you test it with iodine, the color that comes out is blue-black. So that tells you already that C and D cannot be options. Does that make sense? C and D cannot be options. Again, using the elimination method, because that method will save you time. Then now we go in and we look at the options we have left. So if we add iodine solution to this reagent, this suspension of starch, we can get black. Our Benedict solution might be green because, again, we don't know how much is in there. And our Biuret reagent, the reason, should this be purple if we're testing starch? Are there proteins in starch? Should purple be a positive outcome if we test with Biuret and we're testing starch, the breakdown of starch? Just put yes or no in the chat and let's see. No. So now this is another thing I want to draw your attention to, that sometimes you might find in the mark schemes that the answer says something else. But in this case, purple should not be an option, okay? Don't have proteins mixture, and that leaves you only with B. And if you check the mark scheme, the answer might be different, but this would be the correct answer. Usually students can ask for a paper to be remarked, and sometimes if CIE has made an error in choosing an answer, they would actually issue that as part of the examiner's statement to say, oh, there was an error here, so that is the problem. And so that leaves you with B because the protein test will come out negative, while the iodine test and the Benedict's test should come out positive. All right. I hope that's clear to everyone. Okay, let's look at another one. I'm going to zoom out a bit here so that you can all see the options. But here it says a student carried out a Benedict's test on several different known concentrations of alpha glucose. Which graph represents the results correctly? So Benedict's test several different known concentrations of alpha glucose. So here it says the concentration of alpha glucose is increasing here on the x-axis, and the time for the color change is here on the y-axis. So if there's a high concentration of glucose, will the time for color change be faster or slower? I know this one looks a little bit confusing. It seems everyone is a little bit lost. Okay, the concentration of alpha glucose is high, then the time for the color change would be low, which is what is being depicted here. Okay. That concentration of glucose is increasing, okay, and that means, so let's say, for example, over here, the concentration of glucose is 10, and let's say over here it's 5. If you were to trace the time here, I'm not great at drawing. I'm sure if you've been watching my videos, you've already learned that about me. And it's even worse when you're trying to draw with a pen and a mouse or whatever it is you might call it. Okay, so here, let's say this is 10. If we trace this to the y-axis, let's assume that that's absolutely correct, then this might be, let's say, 2 seconds. But if you look at the value for 5, it's a lot more. That's not accurate, but it's basically more than what 10 would be. And in this case, I kind of started on the correct answer, which is A. The answer here is A, but I'm going to show you why the other ones can't be correct. First thing you must pay attention to here is the differences in the labels of the axes. Now, because you look at A and you see that, oh, it says time to color change concentration of glucose, is not the same thing across all the other options. So when you're looking at the other options, you can't get confused because some of you might have looked at this and said, oh, these two are the same, but they are not the same because the axis labels are different. This one here is saying as concentration of glucose increases, the time for color change also increases, which means at a low concentration of glucose, which is here, there is a low time for color change, which is not correct. So that's why B can't be the answer. And if you look at C, concentration of glucose increasing, time for color change is going in a way that it starts to bend over. These graphs actually genuinely do not apply to color change of glucose in any way. So you're left between these two, and B obviously can't be the answer. So I hope that's clear. If it's not, again, just post it in the, what's it called now, in the chat. And I'm sure at the end of the live, I'll be able to go back and look at some of your comments and address some of them. So please don't be shy to ask any questions that you might have, all right? I don't want us to stay for too long so that we don't get too tired, because I know students just get bored and start leaving. Okay, let's look at this one. The diagram shows 3 hexose sugars, and it says which row correctly shows examples of carbohydrates in which these 3 hexose sugars occur. First of all, what sugar is this one? I'm going to call it number 1. This one's number 2. This one's number 3. What is the name of number 1? Oh, that's been done here already. Clearly, I'm the one who's not paying attention now. What is the name of number 1? Just put it in the comments. Okay, I see a request to repeat question 8. So let's just go back to question 8 very quickly in that case. All right, let's look at question 8. So again, let me quickly go over this again. I'm just going to erase some of these. So here, the question is asking that if you test different concentrations of alpha glucose with Benedict's, what would your results look like? And the results they are trying to identify is the time to the first color change. So basically the time it takes from when you add the Benedict's and you boil the solution. For those of you, I hope you all know your food tests by now. So from the time you start that boiling process, how long does it take for you to see the first color change? All right, against the concentration of glucose. And the big question here is, if glucose concentration is high, then the time for the first color change will be low. Because again, think about it, there's a lot of glucose. So when you add the Benedict's and you put it on the stove or the Bunsen burner to boil, the color change will happen faster because the glucose concentration is high. All right. So this tells us that this here is a possibility. If we go and look at B, B says the concentration of glucose is increasing, but as it increases, the time for the color change is actually is actually high, sorry. That's what it's actually saying here. Because if you look at it here, so now it says time to first color change, concentration of glucose. So let's try to do an example here. So I'm just putting random numbers in. Just going to erase there and say this is maybe 6. Okay, so here, and then this is maybe 2, 4, 6. I think this might actually be indeed confusing. And oh, just by the way I teach my students, I always encourage them to offer explanations, because sometimes I look myself and I'm like, oh, wait a minute, what's happening? So please, by all means, if you feel like this is getting a bit weird, just say so. So now let's look at this here. This here, it says the concentration of glucose is, let's say, an assumed value of 2, and we can just sort of assume that the time for the color change there would be 6. And in this case, if it's 4, then time for the color change would be less, and if it's 6, except we can't read a graph from the y-axis, we need to read it from the x-axis. So this here is telling us that at a color change, at a time of 2 minutes, let's say, for example, that the concentration of glucose is around 2. And this over here, so we can't read, you can't read from the y-axis down. You need to read from the x-axis to correspond to the y-axis. And that's because the x-axis, I believe, is the dependent variable. So you need to take it that way. So here the color change is 4. These are my assumed figures, by the way. And it's telling us that if the time for the color change is 4 minutes, then the concentration is, let's say, 4. Let's just assume. And if the time is 6, but basically this makes no sense. Okay? And I'm going to explain it to you again so that it's clear, because I just did some thinking out in my own head. All right, let's look at this again. So let's say this is 2 minutes. Let's say this is 4 minutes. And let's assume this over here is 6 minutes. I'm going to drag it out a bit more. Let's say this is 6 minutes. Okay. This is telling us that when it takes 2 minutes for the color change to happen, our glucose concentration here is about, let's say, 4. Let's say this is 4. When it takes 4 minutes for the color change to happen, our glucose concentration is about 2. When it takes 6 minutes, our glucose concentration is even less than that, which would make sense. But for some reason, this is not the right answer. And this makes more sense. I'm assuming the reason why this is so is because of the arrangement of the axis. But 8 should be number, let me just be sure. Sorry, I'm doing some thinking right now as we look through this. Let's look at C, because I think that might help me a little bit more with understanding why this answer should be A. So C over here says concentration of alpha glucose is increasing, and then the time for the first color change. So if the concentration is 2, the time is, I'm just doing this as a way to make it clear. So if you look at this here, as the concentration increases here, so does the time for the first color change. So that's what makes this one wrong. Okay, because as concentration increases, the time to the first color change should be decreasing, which is what we have seen with A and B. And obviously here, the concentration as it increases, the time to the first color change is also increasing. So that makes D impossible. I hope that's a little bit clear for everyone. I see some of you already on trying to make sure we clarify what's going on here. And so with A, so we either have A or B as our possible answers. And if we look at this here, the time to the first color change is increasing, and concentration of glucose, the concentration is less, then the time to the first color change is actually higher. All right. And as the concentration increases, the time for the first color change is less, which is similar to what we're getting here. But again, time to the first color change here. Okay, I'm not going to dwell too much on this question. I think we can park this question and I'll try to do, I'll do a video about it to explain it because I don't want us to dwell on it too much. But the correct answer is indeed A. I just need to figure out why B is confusing. But C is obviously not the right answer because it negates what our principle is. And D also can't be the right answer. So we know that we are stuck between A and B. And A is the right answer for this question. But I just need to think through why B cannot be the answer. And I think what I'll do is I'll post it in this live, like as a comment when we're done, so that everyone can see how we thought through it, or I can explain how I thought through it. But I don't want us to dwell too much on it for now, so that we don't waste time. And also just let you know that this actually happens in my classroom a lot, where we all walk through the problem, and sometimes we have to take the problem home and bring it back before we can get the solution. So don't be disheartened by that. Just know that some of the questions can get quite confusing for anybody at all. All right. So I see most of you have already guessed correctly that number 1 is alpha glucose. Number 2 here, I'm not going to waste time, is beta glucose. The way you can tell the difference between alpha glucose and beta glucose is to look at this carbon here. So whenever you're given the ring structure, the way to number the carbons is to go 1, 2, 3, 4, 5, and 6. All right, so those are the 6 carbons in glucose. And you know that glucose has a formula of C6H12O6. All right. Now, with alpha glucose on carbon number 1, the hydroxyl group is at the bottom, while with beta glucose, the hydroxyl group is at the top. Okay, so this is beta glucose. And this here, the one that looks like a house, so I usually tell students it looks like, you know, when you draw those house structures or when you used to draw them when you were a kid. Well, I don't know how you guys draw these days, but when I was a child and I was told to draw a house, this is what I drew. So I always imagine that the fructose looks like the roof of a house. And this is beginning to look like a face, but that's not the intention. It's just I don't know how to draw at all. I'm just really bad at it. So this here, this ceiling type or roof type structure here is what you see here. And that is how I identify that this is fructose. Okay. So now the question says here, which row correctly shows examples of carbohydrates in which these 3 hexostructures occur? In sucrose, what makes up sucrose? Does everybody know? What's the formula for sucrose? What do you add together to get sucrose? Just give it your best shot. I hope you remember it though. Sucrose is glucose plus fructose. Okay? Sucrose is glucose plus fructose. And then cellulose is made up of beta glucose. All right, so cellulose is made up of beta glucose. And starch is made up of alpha glucose because alpha glucose is what makes up amylose and amylopectin. So in this case, if we were to look at this, we would then say, because you have to pick just one, right? One of each for each thing. In this case, where it says sucrose as alpha glucose, you would eliminate those out there. I hope that makes sense. Sucrose does not have beta glucose. It actually has alpha glucose. So again, you eliminate that. And then you're left with D as your answer. Sucrose has alpha glucose. Cellulose has beta glucose. And if you look at this other option, let me just show you again why C cannot be correct. If you look at C again, it says cellulose has fructose, which it doesn't. So that can't be the answer. All right, so you can actually just look at it that way, and that negates this. That's also another reason why you can say B is also not correct. And you can say, maybe I'm choosing between A and D, but if you look at A, it says starch has fructose, which it doesn't. So you're left with only D as your possible answer, and that's why the answer for number 9 is D. I hope that makes sense to everyone. Okay. Okay, let's move on. Let's look at number 10. By the way, when you do this paper in the exam from next year, for October, November, you only have 1 hour. So if you're doing this exam in October, November, this paper takes 1 hour. So you have 1 minute, 30 seconds per question because you have 40 questions. So please use your time wisely, but also don't be in a rush. Because if you rush, you might think, oh, I've chosen the right answers, but you might be wrong. So make sure you take your time, use the elimination method as much as you can so that you can select the right answers. So let's look at question 10. It says, what can occur during condensation of carbohydrates? What is condensation? Let's start there. What is when we say condensation, what does that mean? Oh, I see that I've just opened I sort of shut it down a bit. Can everyone see it? Sorry about that. What is condensation? I'm just going to zoom in so that the questions are clearer. If you can define condensation in the chat, just a brief sentence. Release of water, losing of water, that's good. So whenever you have two monomers coming together and there's a release of water, as in the formation of a polysaccharide or a disaccharide or a dipeptide or an amino acid chain, a polypeptide chain, that is a condensation reaction. Based on that understanding then, what is the correct answer for 10? I think it's a pretty obvious answer. It's 10. So if you have a condensation of carbohydrates, would you have a disaccharide produced? Would you have the breaking down of glycosidic bonds? Would you have the use up of water molecules? Or would you have the formation of monosaccharides? The answer is A. You are all correct. The answer there is A. Whenever you break glycosidic bonds, that is not a condensation reaction, that is a hydrolysis reaction. And hydrolysis is the opposite of condensation, okay? So in hydrolysis, you don't release water, you use water. This also then tells you that this is hydrolysis. And when you have hydrolysis where you break bonds and you use up water, you will produce monosaccharides. So the only possible answer here is A. Let's look at 11. That was nice and quick. What is true about triglycerides? So again, I just want to ask, what do you remember about triglycerides? What do you think they're made from? What do you remember about triglycerides? Anybody? Triglycerides? What do you? Fatty acids and glycerol. 3 fatty acids. That's why they're called triglycerides. And you know that fatty acids are hydrophobic. Okay? Fatty acids don't like water. So whenever there's water, they shrink away. Now, if you look at these options that you have, and again, this is the method I always use so that I don't waste too much time looking at option A, okay, B, okay. I just say which option is not going to be the answer so that I don't waste my time looking at it. And the option here, hydrophobic, it says here that they're not. So C and D cannot be options to consider because it's telling us here that triglycerides are not hydrophobic, which is not true. They are hydrophobic. So it's either A or B. Then it says insoluble in alcohol. That's not correct. The way you can dissolve a fatty acid is to actually put it in alcohol. If you put it in water, it's not going to dissolve. So if you're at home right now, just take a glass of water and put a little bit of cooking oil on it and mix it together as much as you can. What you will notice is that the oil comes straight to the top after a while and just sits there. It doesn't move and it doesn't dissolve in the water. But if you take alcohol, so maybe if you have a pharmacy grade ethanol at home, they call it rubbing alcohol. You can use it to make your own deodorant, for example. Like if you want to make your own deodorant, you can actually use it to do that. If you take that grade of alcohol and put a little bit of fatty acid or cooking oil in it and mix it, you will find that the fatty acid breaks down, okay, into the alcohol. So it is not insoluble in alcohol, it's soluble in alcohol. So A cannot be the answer, which leaves us with B as the correct answer. Well done. Up some steam now, I think. I think we've hit our good speed at this point. So let's just move on. Well done to everyone. I hope that you guys are finding this helpful, and I hope that this is sort of helping you think about the things that you know and the things you need to go and revise. All right. So I'm very excited for this. Let's see. What is the minimum number of carbon atoms in an amino acid? Now, when it comes to amino acids, I have a way of helping my students remember the structure of amino acids. And I call this the marriage between carbo and amine. And I know some of you are like, wait, what? So let's look at it this way. We have a carboxylic group in the amino acid, okay? And we have an amine group. This is the carboxylic group. It's a carboxylic acid group. And we have an amine group. I call this carboxylic group carbo. So carbo is a guy, male. And I call this amine, and amine is female. Carbo and amine are married. So we are going to draw them in a marriage for the amino acids. All right. So this is amine over here. And she is married to carbo. And because diamond rings are made from carbon, so they have a diamond ring, which is carbon in their midst. And this is carbo. Okay. Now, if you're a religious person, or even if you're not a religious person, every time there's a wedding ceremony, there's someone who does the joining together of the couple. Okay. And again, you can use any other names besides carbo and amine. You can even choose to make them both female. I'm just using this as a generic example. All right. Or you can choose to make them both male. It's totally up to you. But when you have a ceremony, besides the diamond ring made of carbon, you also have the person who joins them together. So you can call this person any name you like. He can be Hamed, he can be Herbert, he can be any name. He can be Hercules, whichever name you think of that represents age. That's the person joining them together. But there's a side chick in this relationship, and that is the side chain. And the side chain can be anything. The side chain can be another amine, because maybe carbo likes two amines. Okay? The side chain can be a sulfur or some other type of molecule. The point is there's a side chain. And this is how my students remember the structure of an amino acid. Now, if you know this structure, I hope you all found that helpful in thinking of amino acids and how to remember them. This is how my students do it in class. So every time we have an amino acid question, the first thing is, who's amine married to? Carbo. And carbo, they're joined by a diamond ring. There's a side chick, and there's that guy who joins them together, or lady. So it can be Helen if you want to use that name. So based on this, what is the answer for number 12? Minimum number of carbon atoms in an amino acid. What is number 12? That should be easy and straightforward. Good. The answer is B. All right. There are two carbons are the minimum number of carbons you can have in an amino acid. Okay. Obviously, if you have a side chain, that side chain can have a carbon. It can be something like that, even like a CH3 molecule. That doesn't matter. This is the minimum number, and that is 2. All right, let's look at number 13 very quickly. If you would like me to go all the way to, if you'd like me to go on for the next hour, can you please put a yes, no, in like yes in the chat? And if you don't want to go on for the next hour, so I'll just count the yeses and nos and decide if we can finish the entire question. So if you're okay with just going on for another hour, I'm happy to do that. If it gets us further in this paper. So just a yes or no. Okay, I see yeses. Okay, I got a couple of yeses. I'm assuming that if you can't stay on, you'll be able to watch the video later on. So, okay, I'm seeing all yeses. So let's power through. Let's go on. All right, let's go on. So let's see. In an investigation, the same concentration of the enzyme phosphorylase was added to different concentrations of glucose phosphate and incubated at 30 degrees Celsius. At 1 minute intervals, 1 drop of the reaction mixture was removed and added to a drop of iodine solution on a white tile. The diagram shows the results of this investigation, so I'm going to zoom out a little bit so that everyone can see. Okay, so here we have concentration of glucose increasing, obviously, you can see it there, and then time increasing downwards, and it shows us the color. So over here, these are brown, the light gray ones are brown. So that tells us that there is no starch. Okay, or let's just say, let's not say starch, let's just say it's negative. Because if the iodine solution doesn't turn black, then that means we have a negative result. And where it turns black, we have a positive result. Okay, then what does the question ask us? The question says, what explains the trend? Phosphorylase catalyzes a reaction converting glucose phosphate to starch. The maximum rate of reaction is reached at 20 milligrams per dm cube of glucose phosphate. We can check that. Substrate concentration is limiting at concentrations of glucose phosphate 25 milligrams per dm cube or less. Enzyme concentration is limiting at concentrations of 25 milligrams per dm cube or less. What do you guys think the answer is here? I don't want you to be too stressed about it. So let's look at each option. So in this case, we can't do an elimination because everything is a bit overwhelming. We've just read through a lot of information, basically. So we need to work on each option and see what is really happening. All right. So it says here, let's look again at the question. We have enzyme phosphorylase, and it was added to different concentrations of glucose phosphate and incubated at 30 degrees Celsius. And at 1 minute intervals, so let me draw the picture for you. We take a tub, we take maybe like a jar, let's say a test tube. I don't have one here now that I could use, but let's say I'm holding a test tube. In this test tube, I have glucose phosphate, all right? And there are different test tubes because it says different concentrations. And I add phosphorylase to all of these different concentrations. So I have test tube number 1 here, 2, 3, 4, 5, 6, 7, 8. 8 test tubes. In this test tube, there is no glucose phosphate. In this one, there is 5 milligrams per dm cube of the glucose phosphate, and it goes on. So you understand that. Then I go and I add phosphorylase to each of these. Phosphorylase is an enzyme. Anywhere you see A's, that tells you you're dealing with an enzyme. I add phosphorylase to each of these. And then at 1 minute interval, so it means every single minute, I go and I take a sample. And I test that sample with iodine to see if I will get a black color. But obviously, if we look at this side here, the concentration of glucose phosphate is 0, so this side must not be positive at all because there is no glucose phosphate. So it's basically just phosphorylase and iodine, so it can't turn black. So that's that. And then 5, we go on like that, and it goes on. So what is the trend in the results? This is where English becomes important. If you look at option A, it says phosphorylase catalyzes a reaction converting glucose phosphate to starch. That would be correct because iodine tests for starch. But the reason why it can't be the answer is because it is not a trend in the result. Does this make sense? It is basically just telling us that there is a reaction that's happening. But the question says, what explains the trend in the result of this investigation? So when you're looking at the trend and the results, it's not speaking about whether or not there was a reaction. It's asking you what happens when you add iodine. That is the actual investigation. Because remember, glucose phosphate was added to phosphorylase in all of them. So that's not a trend. But as time goes on, something is changing. Because look at it. If we want to speak in terms of trends now, Here, zero concentration, obviously at 0 minutes, there's nothing, right? There's no color change. At 1 minute, we start seeing color changes where glucose concentration is high. And then as time goes on, we start seeing more color changes as glucose concentration increases and as time increases. So you're looking at the trend of the effect of the iodine solution or the color change, and not necessarily at the fact that phosphorylase is catalyzing a certain reaction. That is why A is not the answer. And I know for students, this is like, wait, what? But something else I need to tell you about CIE biology is that they use English to confuse you. So you always need to ask yourself, what exactly are they asking me? The trend in the results. What is the trend I see? I see dark colors as I go this way. If I'm seeing dark colors, that means I'm not looking at whether or not starch is present. I'm looking at the rate at which starch is detected. Does this make sense to everybody? I hope you all get that. I'm just going to go on a whim and say, yes, everybody is getting this. So now let's look at our options. So we know that A can't be the answer anymore. Then it says the maximum rate of reaction is reached at 20 milligrams per dm cube of glucose phosphate. So this is 20. That's obviously not possible because over there you can see that there are reactions that have a faster rate than 20. 20 only changes at 5 minutes, whereas at 1 minute we already had changes at 30 and 35. So again, B can't be the answer. Then let's look at C. Substrate concentration is limiting at concentrations of glucose phosphate of 25 milligrams per dm cube or less. And then enzyme concentration is limiting at these same concentrations. So when we look at substrate concentration being a limiting factor, what that means is that there is no more glucose phosphate that's added to the solution, which obviously we know that there is no more glucose phosphate that goes into the solution at all. All right. So definitely it's going to be limited. Okay. We know that there is a certain amount of, let me rather explain it from the enzyme perspective so that it's a bit clear. Let's start with D. D, by the way, is not the right answer, but I'm going to explain why that is. So we have these test tubes. This drawing thing, I don't know. I never knew in my life I would need it as a skill. So let's say we have these test tubes over here. Okay, those are our test tubes. And in our test tubes, we've put in glucose phosphate and we've also put in phosphorylase. Okay, so we've put phosphorylase in each of these test tubes, right? And we obviously put in a fixed amount of phosphorylase. Okay. And we said that as time goes on, when we test a solution from this with iodine, we start getting these positive results here. So obviously the highest concentrations are the ones that start turning black first. But as we go on, even to the lower concentrations, we still get black. What that tells us is that phosphorylase is not limiting. In other words, it is available in enough, like it is available at enough concentration to continue catalyzing the reaction, even though time is increasing. I don't know if that makes sense. So even though we have time going on, phosphorylase is enough that even at low concentrations of glucose, it still catalyzes a reaction that turns glucose into starch. I hope this makes sense. I hope everybody's following so far. So it's not a limiting factor. What is actually limiting in this case is the concentration of the substrate, which is glucose phosphate itself. And the concentration of the substrate, you can see, plays a role because look at this here. Between 0 and 7, for example, there was no positive result for the concentration that was 5 milligrams per dm cube. Okay? Between 0 and 6, you didn't get a positive result for 10. So it means that the substrate concentration was affecting the way the reaction was occurring. Does this make sense? So if the substrate concentration is limiting, it means the substrate concentration is in an amount that is slowing down the rate of the reaction. That's what limiting actually means. And if you've done photosynthesis, I'm sure you've done limiting factors then. So if the concentration of glucose is limiting, that means it slows down the rate of the reaction. And that is just evident when you look at this, that when you're dealing with lower concentrations, the rate of the reaction is less than when you're dealing with higher concentrations. Because look at this, at higher concentrations, by the very first minute, you have results. The reaction is happening. By the second minute, it doesn't stop, it keeps going. So these are not limiting. At 30 and 35, the substrate concentration is enough for the reaction to just happen at the rate that it should. But when you're dealing between 25 and below, so look at 25 here, you get your first positive result after 3 minutes. So that means that the concentration 25 milligrams per DMQ actually slowed things down. Okay, and then you look at 20 and it's even slower, and you look at 15 and it's slower, and it continues to go slower as the concentration of the substrate decreases. So that tells you in this case that the substrate concentration is the limiting factor. Does this make sense to everybody? I really, really hope it does. I know that things with substrate concentration can be a bit weird, and I deliberately chose this paper, by the way, because of how unconventional the questions are looking. Usually they would give you a graph and say, choose which one represents limiting concentration of the substrate or whatever. But in this case, they've given you a reaction to look at. So I hope that it's a bit clear. Again, if it's not, just leave a comment. Once the live is over, maybe sometime later in the evening or tomorrow morning, I will go into the chat and just explain things in more detail that are not clear. And I haven't forgotten that question 8 stumped us a bit, so we might have to go back to that one. So, but I hope that this makes some sense. All right, let's look at number 14. What is the definition of the Michaelis-Menten for an enzyme? Does anyone know what this one is? I think it's a bit easy. I'm just going to draw it here. So when we have this, and over here is Vmax. I'm just going to write that as Vm. And let's say this is half Vm. Okay. And obviously over here we have substrate concentration, and half Vm is the one that corresponds to the Km value. I hope everyone gets this. So based on this, what is the right answer for 14? And also, if you can you just put in the question what you think Vmax, what you think Km indicates, because that's also usually another question that you get. And you're all right when you say the answer is D, because that is correct. But what does Km represent? Can we just look at that quickly, just if you can? Everyone's so quiet. Vmax is the maximum rate of the reaction. That's correct. Km shows you the affinity, all right? Shows you the affinity between the substrate and the enzyme. So if your Km, if your, what's it called now? Your enzyme has a weak affinity for the substrate, your Km value would be higher. Excuse me, which means that you would need a high substrate concentration to be able to reach half Vmax. I'm going to just represent that for you very quickly so that it's not confusing. So let's say, for example, how do I explain this? So let's say, for example, this enzyme has a high affinity for the substrate. It means that at this concentration of the substrate, which is a bit lower, right? The enzyme is able to reach half Vmax, and Vmax is obviously not too high off, not too far off. But if the affinity was less, the graph would look something a little bit different, like maybe something like that. And maybe half Vmax might be somewhere here. This is not a good drawing. Again, I'm terrible with drawing. I think I say that enough. But if the affinity was less, we can say maybe Vmax is somewhere there, and half Vmax would be here, which means your Km would be a lot higher. All right. So what that means is that you would need a higher substrate concentration to reach the half Vmax for this graph, which has its Vmax somewhere up there. I hope that's a little bit clear. If it's not, watch the video on enzymes. I explained this in that video. And for those of you who might not know, all the videos are arranged in playlists. So you can just go to the playlist, click on chapter 3, you will find enzymes right there. All right, let's look at this one quickly. The graph shows the rate of entry of glucose into a cell, shows how the rate of entry of glucose into a cell changes as the concentration of glucose outside the cell changes. So you can see concentration of glucose outside the cell is increasing, and the rate of entry of glucose obviously is also increasing. What is the cause of the plateau at X? What do you guys think the answer is? So concentration outside is high, and obviously that means that more glucose is getting into the cell, but at some point there is a plateau. Why do you think that is? So you can choose either A, B, C, or D. What do you think the answer is? For number 15. Okay. I see Fatima is answered. Does anyone? Okay, I don't want to waste too much time, so I'll explain this so that we don't stay here for too long. E, because here it tells us on the graph that the concentration of glucose outside is increasing. So if the concentration of outside is still increasing, this is the plateau. So the plateau is the flat part. Okay, so this flat part of the thing here, that's the plateau over there. So if within the plateau we still have an increasing concentration of glucose, that means that the glucose outside and inside are not the same. Now, B says the carrier proteins are denatured and no longer able to function. There is nothing in the question that tells you that that has happened. Nothing in the question says there's a high concentration or the pH has changed or anything like that that would affect the structure of the protein. The cell has used up its supply of ATP. Again, you don't even know what kind of cell this is. So you can't make that conclusion. All the carrier proteins are saturated with glucose? Yes, that is your correct answer right there. It's A. The reason it's A is that look at it this way. When glucose, so let's say this is the cell over here, and this is outside, and we have all of these glucose molecules here on the outside. Okay, so at first we start with a few glucose molecules, and there are all these channel proteins on the surface of the membrane that are happy to bind with the glucose molecules. All right, so all the glucose molecules at first will find proteins to bind with. They come in, there's a protein for each glucose molecule, so they're very happy. Okay, so they all bind, they all pass in, and obviously more glucose starts increasing. So as these are binding, there's now an increase of glucose that's happening on the outside, as the graph shows. So it will get to a point where the glucose on the outside will be so large in quantity that the proteins that are available to transport them into the cell are not enough. So it gets to a point where the proteins have to, because the proteins say they bind one glucose at a time. So they have to bind this glucose, transport it into the cell, and then come back and take another one. Meanwhile, the concentration on the outside is increasing. So it would reach a maximum rate, which shows that all the proteins have now been saturated. So it means that's the best rate you can ever get, and it doesn't go higher than that. I don't know if this makes sense to everybody. I can try to explain it maybe using, let's say, your school hall. All right, so let's assume that at your school there's a party. Okay, there's a party at your school and you are 1,000 students in number. Now, the hall where this party is happening has only 2 doors. You can only go in through 2 doors, and each person, the doors only allow 1 person in at a time. So think of those doors that open, you go in, they close, they let you out. Okay? So there are 1,000 students coming to this party. The first 2 students who get to the party will be able to go through the doors without any problem because, you know, there are not many people. So they go in, the doors open, they go out they go into the hall and they're in the party. But as those 2 students are going in, 4 more students arrive. Now the reaction is still okay because 2 students, okay, 4 more, let's just go 2, 2. But at some point, if 20 students arrive at the same time, the doors do not increase in number. The number of the doors stay the same, and only 2 students can go in. So even though the concentration of students on the outside is increasing, the rate at which the students will be able to go in cannot increase because the number of doors to the hall have not increased. Does this make sense? I hope it does. I know that the network might be a bit slow, so I can't see all your chats in real time, but I hope that that helps you understand it. So that is why the answer there is A. All right, we're almost halfway. Let's look at 16. 16 says the diagram shows the movement of a substance Z across a cell surface membrane. And this is Z, you can see there. It starts off in a vesicle, and then the vesicle merges with the cell membrane, and then Z is released outside the cell. Which process is involved in this movement? Come on now, this is an easy one. Is it A, B, C, or D? So is this going to be A, is it B, or is it C, or is it D? Come on, give it a try. Yes, I see B. That's correct. Yes. Yes, that's correct. So the answer here would be B. Endocytosis is the swallowing. So endocytosis is things being taken in by the cell. Exocytosis is things being released by the cell. Now, phagocytosis is an example of endocytosis. All right. So make sure you know that. Phagocytosis is an example of endocytosis, and it involves digesting solid material. While pinocytosis is also endocytosis, but it deals with liquid material. Okay. So that one's quite straightforward. Let's look at number 17. It says here, a Viking tubing is often used as a model during experiments to investigate osmosis in plants. So I hope you guys have done this practical at your school. Please, if you haven't, by all means, ask your teacher to explain it to you so that you understand it, because you get questions like this a lot in CIE. What could the Viking tubing represent? But now let's say you haven't done this experiment. I want to ask you a question. What is the definition of osmosis? Just put it in the chat. What is the definition of osmosis? Come on, quickly. Everyone's quiet. Okay, I will assume that it's the network that's a bit slow, but osmosis is the movement of water, okay, from a region of higher concentration to a region of lower concentration across a semi-permeable membrane. Osmosis is not what happens across a cell wall. It's a semi-permeable membrane, and in this question, the 2 semi-permeable membranes you have are the cell surface membrane and the tonoplast. The tonoplast, for those who might not be clear, is the membrane of the vacuole. So it's a membrane as well. All right. So already you know, because osmosis doesn't occur across the cell wall, A cannot be your answer. D, also cannot be your answer. So you're left with B and C. And I think Fatima already told us the answer, that because this is a membrane, osmosis can also happen across it. And that's what the Weiskin tubing is used to show. And so the answer here is B. All right. C can't be the answer because it's saying that it can't be a tonoplast. So the answer is B for number 17. Let's look at number 18. A scientist stains the chromosomes of a diploid plant cell with a fluorescent dye to observe the telomeres. The cell has 38 chromosomes. How many telomeres will the scientist observe in one of the nuclei during telophase of mitosis? There are 38 chromosomes. This is in telophase. Remember, in telophase, the cells are already splitting into two. They look something like that. Okay? Now, in each one, so it says the cell has 38 chromosomes. Obviously, in mitosis, you have the same number of chromosomes in each cell. So if there are 38 chromosomes, how many telomeres will there be? Is it A, B, C, or D? Is it A, B, C, or D? If there are 38 chromosomes, how many telomeres will you observe? B, that is correct. Because remember that when you're dealing with telomeres, you're dealing with the edge of the chromosome. So I'm just going to draw a chromosome here very quickly. All right. So there you are. That's what you're dealing with. So this over here, sorry, something's in my eye. Okay. So this over here would be the telomeres, but this would be considered the telomere for one chromatid and then the telomere for another chromatid. So you just have to multiply 38 times 2 and you get 76. Okay, so that's the answer for that one. Just going to erase this. Okay, this is the photomicrograph. I love mitosis questions, by the way, because I think they're just really easy and straightforward. The photomicrograph shows a cell during mitosis. What is happening in this cell? First of all, what stage of mitosis is this? If you look at this micrograph, can anyone just give an example? What stage of mitosis are we looking at here? Is this prophase? Is it metaphase? Is it anaphase? Or is it telophase? Which my students refer to as PMAT. That's a way to remember it. Prophase, metaphase, anaphase, telophase. What phase is this? Anaphase. Well done. That's correct. This is anaphase. And what happens during anaphase? The reason why I'm taking you through this route to answer the questions is not because I'm just trying to be a pain in your head or anything. I'm trying to get you to think of other ways of looking at the question before you even start thinking of options, to say, what am I looking at and what happens during that phase? You know that during anaphase, do the centrioles replicate? No, that's not what happens in anaphase. Do the spindle microtubules shorten? Yes, they do. Do the chromatin condense? No, they don't. That leaves you with 2, and your answer is C. That's just easy and quick. All right. Let's look at 20. This is a question students hate a lot because they say it's confusing. But I see it a lot, so I hope that we can answer it. Which events are part of mitosis? Interphase, prophase, cytokinesis. What is the answer for 20? What's the answer for 20? Is it A, B, C, or D? Which one is part of mitosis? So I see people saying D, that's correct. Now, interphase is not part of mitosis. Interphase is part of the S phase, or it's made up of the S phase, sorry, the S phase, the G1 phase, and the G2 phase in cell division. Cytokinesis is also not part of mitosis. It happens after mitosis. So your answer here is D. Let's look at number 21. You might see that I'm going a bit quicker. I want us to be done latest by 10 minutes. So in the next 35 minutes, what am I saying? 25 minutes, sorry. I want us to be done with this paper, so I don't want us to waste too much time. But again, if there are questions, post them in the chat, all right? The graph shows the length of the spindle fibers during mitosis. Which region of the graph shows when all centromeres have detached from the spindle fibers? So this is the length of the spindle fibers. At A, you can see the spindle fibers are growing. At B, they reach their maximum. At C, they start to shorten. And at D, there are basically no spindle fiber lengths occurring. So which would be your answer here for 21? I think it's pretty obvious. D, that's correct. Well done. The answer is D because this is showing you prophase. This is between prophase and metaphase. Over here where the spindle fibers are shortening, you're dealing with anaphase because they're getting shorter. They're pulling the chromatids to opposite poles. And then you have D when the chromatids, what's it called? The centromeres or the chromatids and the chromosomes are already at the poles they should be at, and the spindle fibers don't exist. So that would be D. Let's look at 22. A short piece of DNA, 19 base pairs long, was analyzed to find the number of nucleotide bases in each of the polynucleotide strands. Some of the results are shown below. So strand 1, so again, remember in DNA, you're dealing with adenine, cytosine, guanine, and thymine. So in strand 1, there are 4 thymine molecules. In strand 2, there are 5 thymine molecules. Then the question says here, how many nucleotide bases containing C were present in strand 1? Isn't that a fun question? Let me explain this. I don't want us to waste too much time on it, so I just want to explain it quickly. So you have 19 base pairs long, 19 base pairs, all right? So that means there are 19 of all of these things added together. It tells you here that there are 4 thymine molecules, so 4T. And you know that thymine binds to adenine. So if there are 4T, there has to be 4A. Does that make sense? Okay, so that then tells you that you're dealing with 19 minus 8. And that should give you, wait a minute, I think I'm making an error here. It's 1 DNA, 19 base pairs long. So if you're dealing with 4A here on the one strand, oh, so it's 1 DNA molecule. Sorry, I think I'm making a mistake here. So 4 here, 5 there. This should be 5 as well. So 7 here, this would be 3. Sorry, that should be 7. Sorry, I think I'm making a mistake. But please, if there's anyone in the chat who can explain it so long, can you do so while I just work it out? Because I don't want us to spend too much time. Okay. The the number of nucleotide bases in each of its polynucleotide strands. All right, some of the results are below. So in each strand, so we have strand 1 over here. I'm just going to try to explain this quickly. If I make an error, then I'll explain it in the chat later because I don't want to waste too much time on this live. I can tell people are getting exhausted and leaving. Yeah, so let's see. So this is a DNA strand, a short piece of DNA, and it has 19 base pairs, okay? So it means that there are 19 bonds that are formed, okay? 19 base pairs means that there are 19 over there. I think these numbers are wrong, so let's just try to work it out. So on strand number 1, we have 4 thymine molecules. Okay, we know that 4 thymine molecules will bind to adenine, so it means that on strand number 2, there should be 4 adenine molecules. Okay, then we also see that on strand number 2 here, we have 5 thymine molecules, and that should correspond to 5 adenine molecules. I hope that makes sense. Okay, so here now, if we look at this, we're now dealing with 4 over here, plus 7, which is 11. 11 plus 5 here is 16. And so that means that if we have 19 base pairs and all of these add up to 16, we then have 3 guanine molecules on strand 2. If there are 3 guanine molecules on strand 2, it means that there will be 3 cytosine molecules on strand 1 because guanine binds to cytosine. Does that make sense? So in that case, then our answer would be B. I hope that wasn't confusing. So I'm just going to go over it again very quickly. We were already given that we have 4 thymine molecules, and we know that thymine binds to adenine, okay, and cytosine binds to guanine. If there are 4 thymine molecules on strand 1, there has to be 4 adenine molecules on strand 2 that will bind to that thymine. Okay? If there are 5 thymine molecules on strand 2, there has to be 5 adenine molecules on strand 1 to bind to that thymine. All right? So once we have those values down, again, if we look at the bottom on strand 1, we then know that, okay, 4 plus 7 is 11. 11 plus 5 is 16, but we are dealing with 19 base pairs. If there are 19 base pairs, we just need to deduct the 16 from it to get the amount of guanine we have on strand 2. If there are 3 guanine molecules on strand 2, they must be binding with 3 cytosine molecules on strand 1. And that is how you get 3. And so the answer for number 22 should be B. I hope that that's clear. Okay, I hope that explains it. This one, it says, which nucleic acid bases are purines? Okay, this is just something you have to learn in your book. So your pyrimidines, which are the PYR, would be your cytosine and your thymine. Those are the pyrimidines. And a good way to remember them is to see that they have Y in their names. And the purines are your guanine and your adenine. And so that would make your number 23C. Let's look at 24. In a genetic engineering experiment, a piece of double-stranded DNA containing 6,000 nucleotides coding for a specific polypeptide is transcribed and translated. What is the total number of amino acids in this polypeptide? I love this question because it's a math and biology question at the same time. So you have 6,000 nucleotides. Okay. And whenever you're reading the DNA, so obviously if you have 6,000 nucleotides of DNA, You're also going to have 6,000 at RNA, just by the way. Okay, and that's the transcription process. If you haven't watched this video, please check it out on YouTube. It's chapter 6. Check it on the YouTube channel. So you have 6,000 mRNA. Whenever you're reading your mRNA and converting it into protein using tRNA and whatever, you read it in threes. So that's why we call it a triplet code. So you just have to divide this by 3 in order to get, is that what you need to do though? So you take the DNA and then you go into the mRNA. Sorry, I've made an error. This is wrong. Wait. You go to DNA, you take the DNA, and then you divide that by 3 to get your mRNA. Because remember, your mRNA is made up of its own molecules. So you're reading it in threes. So you read, for example, A, C, U, G, C, T, and you go on like that. All right, so you read it in threes. So that's why you have to divide by 3 to get the mRNA. When you get the mRNA, you then need to convert that mRNA using tRNA into a protein. And again, that divides it by 3. So that then becomes, I think I'm making an error here. I found my error. Sorry about that. I'm going to explain this again. But if someone has already got the answer, please put it in the chat. But I've made an error. It says a double-stranded DNA has 6,000 nucleotides, okay? Double-stranded is a key word here. When you're going from DNA to RNA, you don't read the 2 strands, you only read 1 strand. So we have to divide this by 2, okay? So it's going to be 6,000 divided by 2, and that then gives us the 1 strand where the mRNA is read from. That's 3,000 nucleotides that would be read and converted to mRNA. Those 3,000 nucleotides will then be transcribed and translated and read in threes using the triplet code. So that then divides this by 3, and that should give us 1,000. And I believe 1,000 is the right answer. Yes, it's here. And the answer there should be B. I hope I explained that well. So again, I'll just go over it again very quickly. If you're dealing with DNA, it doesn't tell us that there are 6,000 base pairs or whatever. It just says there are 6,000 nucleotides, a piece of double-stranded DNA. So it means that when you look at this DNA, you have 1 strand here, you have 1 strand here. 3,000 nucleotides are here, and another 3,000 are here. When you want to convert into protein, the RNA polymerase is going to come and it will read only 1 strand. So let's say it decides to read from here. So it means it's not reading the 3,000 that are here. It's only reading the 3,000 on the one side. So that's why you have to divide this by 2 to get 3,000. Now, when it reads those 3,000 nucleotides, it will read them, it will take them to the ribosome as mRNA, and then the tRNA will come and they will be read in threes. So that means to make 1 amino acid, you have to read 3. Okay, so AUG is 1 amino acid. ACU is another amino acid. So because we are reading in threes, we then need to divide the 3,000 we read from here by 3 to get the number of polypeptides, or number of amino acids rather, that we would have in the polypeptide, and that is 1,000. So I hope that's clear. Again, if it's not clear, just drop your comments. We will try to explain it later. All right, let's look at 25. Which statements about tRNA are correct? So this again is just about learning facts. Hydrogen bonds between bases temporarily hold tRNA against mRNA. That is true. Okay, and again, this is another technique you can use whenever you're answering questions. You can just look at the statements and say which one is true and which one is not before you even go into the options that you have. All right, so that's true for that one. And then the bases in tRNA molecules are the same as the bases in mRNA that is being translated. That's not true. They are the complementary bases. So if you have an A on your mRNA, you will have a U on your tRNA. So they are not the same. tRNA translates the base sequence in mRNA into the amino acid sequence of a protein. That is true. So we have 1 and 3 as the correct ones. That means A, B, and D can't be the answer. Our answer here is C. I hope that's clear. Let's go on quickly. Oh, I was wondering. Okay, so this is a topic I know students hate a lot because they hate transporting plants. But you just have to love plants. You don't have a choice if you're doing CIE. So let's look at 26. It shows the transverse section of a part of a dicotyledonous leaf. In this case, I tell students there is no teacher who can explain this to you in detail. Just study the drawings for the leaf structure, for the root structure, and for the stem structure. Always make sure you study those because those would be the only way for you to be able to ace this chapter. All right. So number 1, let's see here. It says, what are the correct labels? So 1 here is pointing there and 2 is pointing there. Obviously, this seems to be a little bit upside down because I think usually it's the other way around. But in this case, based on what I remember about the leaf structure, 1 is the xylem and 2 is the phloem. So A and B should not be correct. So we're looking at C and D. And then we have the spongy mesophyll and the palisade layer. The palisade is usually closer to the outside here. It has longer cells, while the spongy mesophyll has these kinds of cells. So my money here is on D as the correct answer. But you can always cross-check that just to be sure. Let's look at 27. I want us to end in about 13 minutes. So let's try to push through. Some of the features present in transport tissues in plants are listed. So here we have lignified cell walls, cytoplasm, mitochondria, pits, plasmodesmata. Which of these features are present in phloem sieve tube elements? Do phloem sieve tubes have lignified cell walls? I'm checking that. Do you think they have? I'm going to zoom in. I see that I zoomed out so that you could see the other diagram. But here, do phloem sieve tubes have lignified cell walls? Does anyone know? Come on, come on. So I don't want to waste too much time, but lignified cell walls are only in the xylem. Okay. Do phloem sieve tubes have cytoplasm? Yes, they do. They have streams of cytoplasm. You can call it that. Do they have mitochondria? Yes, they also do. Do they have pits? No, they don't. Pits are only in the xylem. Okay. Do they have plasmodesmata? Yes, they do. All of these cells actually have access to plasmodesmata, so these are also present in the phloem. And so in this case, the answer would be C, all right, for number 27, because that is where you find your phloem sieve tubes. They don't have lignified cell walls. Remember, the lignified cell walls are for the xylem to strengthen the wall of the xylem. That's why lignin is present. But the phloem cells don't need that. So yes, Kalhara, you're correct. The xylem is the one with the lignified cell walls. Let's look at 28 quickly. I'm just going to zoom out. I think my students have seen this particular question before. Shows the relationship between phloem sieve tube elements, xylem vessel elements, and companion cells. Now, I want to tell you guys, whenever you have this kind of question where they draw a Venn diagram that looks like this, wherever the two diagrams intersect, they're basically saying whatever is at number 1 and number 3 are sharing what is in number 2. Okay, and 3 and 5 are sharing what's in number 4. So please just have that at the back of your mind when you're answering this question, because I find that some students who are new to CIE get a little bit confused. Okay, so let's look at 28. So number 1 tells us that we're dealing with companion cells. Companion cells are the cells that are attached to the phloem and they aid in sucrose transport. Then it tells us number 2 is endoplasmic reticulum, and then number 3 is phloem sieve tube element. Now we know that indeed the phloem sieve tube elements and the companion cells have endoplasmic reticulum, so that is certainly correct over there. Okay, so far so good. Then it tells us that number 5 is the xylem element, and there is no nucleus, but also says the phloem sieve elements also don't have a nucleus, which is also correct. Okay, so it seems like everything is correct in A. But let's look at another one just for the sake of looking at it. Let's say we look at C, for example. So in C, phloem sieve tube elements, mitochondria. Well, the companion cells in the attached to the phloem sieve tube elements do have mitochondria, but do they have a nucleus that the xylem vessels also have? The answer is no. So C can't be correct. Remember that the xylem vessels don't have anything inside of them. They're just empty tubes that aid the transport of water. All right. The answer here is actually A for number 28. But I would advise that you spend some time looking at all of these other options to see why they are not correct. Okay. And if you have any questions, you can just post it as a comment and we will be able to look at that. Which statement correctly describes a transport pathway in plant? Okay, so in plants you have the symplast. Let me rather underline than just take my time. You have the symplast and the apoplast pathway, right? And the apoplast pathway, water may move through the plasmodesmata. And the symplast pathway, water may move through intracellular spaces. The apoplast pathway may be blocked by the Casparian strip. That's actually true, because when water is moving from the root hairs and trying to get into the xylem, it gets to a point where it is blocked by the Casparian strip and then forced into a symplastic pathway. The symplastic pathway is not blocked by the tonoplast, so that's not correct. Both A and B, in the symplastic pathway, water may move through the intercellular spaces. I can't really remember this if I'm being honest, but I do know that there's a part where the water moves through the plasmodesmata, and there's another type where the water moves through the cell walls and all of that. So I think I'll just fact check that, but I would go with my answer as C here, because that is definitely a correct one. Let's look at 30. I think we're making good with time. And I know it's a bit rushed, but I hope that some of the tips you've picked up earlier on are helpful. The photomicrograph shows an external view of the mammalian heart. And then which row identifies the position of the structures labeled P, Q, R, and S? So now when you're looking at the heart, if you're not told anything, I always tell students, always imagine you're looking at a mirror image. Okay, so the mirror image. So in this case, if you are facing the heart, if this was not a mirror image, this would be the right side of the heart if you're following with your hand, and this would be the left side. Okay, but once you flip it around, you're now looking at the left side and the right side is on this side, if that makes sense. I hope you guys got that. So if this was the heart, if it wasn't a mirror image, you were looking at it facing front as though it's in a person, then this would be the right side. So my thumb is the right side and my finger is the left side. But if I turn it around as a mirror, which is what you have to assume with this question, then the left side moves to your right. Okay, so in this case, that means that P and Q are on the left and C and D cannot be our answer. Okay, I hope that makes some sense. Yeah, and then P, the left atrium. So the atrium is the one on top. So that's definitely correct for both of them. And then Q is the left ventricle. The ventricle is the vessel at the bottom. If you haven't watched the videos on chapter 8 where I discussed the heart, please make sure you do so. This cannot be the cardiac muscle. That's not it. Okay. Also, porcine tissue, you actually don't see that when you're looking through the heart. Pulmonary vein, well, S could be a pulmonary vein, or it could be the vena cava actually, because it's coming from the top. So in this case, I would say if you were to choose based on what you actually know. They've made it easy by labeling Q as the cardiac muscle, which is not what it is. That is a chamber in the heart, because when you cut it open, you will see that it's a chamber that actually goes all the way down. So Q cannot be the answer there. And so B would then be your only possible option. With these ones, sometimes with Purkinje tissue, coronary artery, well, sometimes they're just hard to figure out based on how the heart is looking here. So I would go with B as my answer. Okay, so that's for number 30. Let's do the last 10 questions very quickly. What happens during the ventricular systole? So systole is a contraction of the heart. Always remember that. Diastole is the relaxation of the heart. Okay. And you always start with the atrial systole, then you do the ventricular systole and the ventricular diastole. That is the cardiac cycle. So again, watch the videos on chapter 8 so that you can follow through or you can understand. So here, the atrioventricular node transmits an electric signal to the apex of the heart. Well, that's not what happens there with the ventricular systole. The pressure in the ventricles drop below the pressure in the atria. That's not true. Again, remember, systole is a contraction. So when you have a contraction, you actually have increased pressure. So here the atrioventricular valves close and the semilunar valves open. That is true. Again, for you to gain an understanding, just go to the playlist, select chapter 8, and watch those videos because they explain this really, really well. I don't want to keep you guys too long. Let's look at 32. Athletes often move from low altitude to high altitude to train for a race. Which statements about the effect of training at high altitude are correct? Okay. So if you go and train high altitude, what happens to you? When you're at high altitude, what happens is that your blood cells, your red blood cells actually increase in number, which means that your oxygen transport might be much better. So if you're trying to run a long race, you want your heart to be able to give you as much oxygen, or your lungs, or whatever the combination of those two are, to give you as much oxygen for as long as possible. So if you're running a marathon, for example, you can't say, oh, I'm just going to run in my yard 10 times a day and then I'm fine. Except you live at a high altitude area, then that would be sufficient. Sometimes you need to go to a place where you really push your heart and your red blood cells. So let's look at this. Which statements would be correct here? This is number 32. So it says higher concentrations of carbon dioxide stimulate greater oxygen dissociation. Well, that is correct, but at high altitude, that doesn't really apply. The air is thin at high altitude anyway. But let's just see. Lower concentrations of oxygen stimulate the uptake of more oxygen by red blood cells. Lower partial pressures of oxygen stimulate higher production of red blood cells. This is the answer here. Whenever people live at high altitude, so for example, I live in South Africa and some cities in South Africa have high altitude. And so when you go running here, for example, it's not the same as running at sea level. When you run at sea level, life is a lot more pleasant. When you run in certain cities like maybe Johannesburg, you might find yourself gasping like, even though you're really fit. And that's because here your body has to get used to producing more red blood cells at a higher altitude. So at high altitude, people have more red blood cells than people who live at low altitude. And so people who live at high altitude and train there, when they go to lower altitudes to go running, for example, they're able to run a lot better than someone who's only lived at low altitude because they tend to have more red blood cells. So that's the thinking behind that. So the answer there is D. Let's look at number 33. We're making good progress. The graph shows the oxygen hemoglobin dissociation curves at a pH of 7.6 and a pH of 7.2. Which point on the graph shows the percentage saturation of hemoglobin in the blood leaving an active muscle? Okay, so an active muscle, will it have high oxygen or will it have low oxygen? I want you guys to just think about that very quickly because I realize I've just been speeding on, like just going on very quickly. Will the oxygen at be higher or will it be lower? And also, will the hemoglobin be saturated with oxygen or not? Will it have a higher saturation or a lower saturation? Just going to check the comments. For 33. Yes, that's correct, Cholera. It will have a low saturation of oxygen. Okay, so when you have a respiring muscle, an active muscle is basically using up oxygen. So by the time you're measuring the saturation of hemoglobin there, it would be less than a muscle that is not respiring. So if we look at here, we would depict an active muscle. It can't be A or C because these two here, again, when I draw, I just go overboard for some reason. I don't know. It's either I underdraw something or I go big or go home. I don't understand it at all. So if you look at these two here, they're actually showing that the saturation of hemoglobin is higher than what's happening down here. So A and C can't be your response. You have to choose between B and D. The saturation of hemoglobin in a respiring muscle would be really low. That's its saturation with oxygen. Okay, it would be really low because, again, a respiring muscle is using a lot of oxygen. So any hemoglobin that's leaving that muscle, again, remember it says in the blood leaving the muscle, the hemoglobin that's leaving that muscle will not have as much oxygen as hemoglobin that's maybe coming into the muscle. So in this case, the answer would be B because it can't be D. D has a higher percentage saturation of hemoglobin. Then let's look at 34. Which row correctly describes the short-term effects of carbon monoxide and nicotine on the body of a smoker? Carbon monoxide, in terms of demand for oxygen, what do you think happens when there's carbon monoxide? Does the demand for oxygen increase or decrease when there's carbon monoxide in the blood? Does the demand for oxygen increase or does it decrease? Come on, I'm sure you guys know this. So I'll give you a clue. Carbon monoxide binds with, thank you, Kalhara. Carbon monoxide binds with hemoglobin and it binds irreversibly. So it means that the hemoglobin doesn't transport as much oxygen as it should, and that then increases the need for oxygen, okay? So already that tells you that A and B can't be correct. Demand for oxygen, it's increased by carbon monoxide, okay? Oh, sorry, it's also increased by nicotine as well because nicotine affects, what nicotine does is that it binds to, like, it causes the blood vessels to constrict. So this is actually more of a nicotine issue than a carbon monoxide issue. Carbon monoxide is poisonous to the body in the sense that it binds to hemoglobin irreversibly. And so that means that that hemoglobin is not able to transport oxygen. Nicotine causes a constriction. So when we say constriction, it reduces the diameter of the blood vessels and obviously increases the need for people to take in more oxygen. Concentration of oxygen in the blood, that would be decreased by carbon monoxide because, again, it binds to hemoglobin irreversibly. And so the answer there should be D. Okay. I hope that was clear. If it's not, I'm willing to explain it once we finish our live session, which we're almost done. Which statement about bronchioles are correct? So, do bronchioles have ciliated cells? Does anyone know? The answer is yes, they do. Do they have goblet cells? No, your bronchioles don't have goblet cells. Goblet cells are responsible for making mucus. Okay, so your bronchioles don't have that because your bronchioles are basically helping with the diffusion of air, okay, to the alveoli. So they don't have that, but they do have muscle tissue. So in this case, again, approaching it this way makes it a lot easier for you because when you then go into the option 1, A, B, C, or D, you can sort of pick out the one that's absolutely correct very quickly. And in this case, the answer is C. Which features are important for the diffusion of oxygen out of an alveolus? Blood pressure forces red blood through the capillaries. I don't know if that's important for an alveolus. Blood pressure forces red blood cells through capillaries. Epithelium is permeable to respiratory gas. That's true. Moist squamous epithelium is present. That's true because that also helps to moisturize the air. Surfactant is present to reduce surface tension. That's not true, actually. So here we have 2 and 3 are correct that we know, okay, that I know for sure. I'm sure you guys probably, because you are writing the exams, you're more proficient than I am at this point, but I've been teaching this for some time. So 2 and 3 we know are correct. Let's now look, and again, this is a tip for you. If you are in the exam and you have options such as these, and you're like, okay, I know this one's correct, I know this one's correct, I know that this one is wrong, I'm not sure about this one. Go and look at what your options are saying. So which options have both 2 and 3? There's no other one. It's only A. So you just take a guess and say, you know what? I'll go with A. I hope that that helps. And I'm telling you this because students sometimes feel the need to always know all the options. But I remember when I started teaching, I observed an older teacher that said, sometimes students need to learn how to rely on guessing properly, not guessing stupidly. So don't go in and play mini, mani, mo, father had a donkey, donkey died. Don't do that. Rather say, what do I know about this question? What am I unsure of? And what do I know is absolutely wrong? Kick out the one that's absolutely wrong. Pick the ones that you know for sure these are correct, and find the options that then give you an idea of what to select. Okay, I hope that that's a helpful tip. And if you use it when you're practicing past papers on your own and it actually works out, please, by all means, let me know. The diagram shows some of the pathogens that cause disease in humans and some of the ways they are transmitted. So this is a very weird diagram with arrows pointing everywhere, but let's not look too much at that. So here it says the pathogen, different types of pathogens. You have a protoctist, you have a bacterium, and you have a mobilivirus, so basically a virus. Method of transmission, you have contaminated food, cough and sneezing, contaminated water. All right. Then what is the correct pathogen and method of transmission for measles? We know for sure measles is not caused by bacterium. That's more of cholera. Protoctist causes, this is cholera and tuberculosis, by the way. They are caused by a bacterium. Protoctist is what causes malaria. Just going to write it as mal. A virus actually is what causes measles, so we know mobilivirus has to be the answer. Contaminated water, contaminated food, those are usually transmitted through, those are things that cause the transmission of cholera in particular. Coughing and sneezing can actually lead to the transmission of measles. And so in this case, the answer I believe would be D for 37. Okay. I hope that was clear in how I worked that out. Which row is correct for each disease? Let's see. Cholera caused by a bacterium. Yes, that's true. HIV/AIDS can be transmitted in breast milk and across the placenta. That's also true. Malaria has an unsuccessful eradication program. Of course, that's true. And all you have to do is just go and live in West Africa, where the mosquitoes seem to have been trained by the military and will follow you everywhere. So you already know that malaria was not eradicated. Measles caused by a virus. So they've sort of given you the answer to the previous question here. That's true. But let's see, with cholera, eating a raw shellfish can be a source of infection. Well, there's no indication or there's no information that tells us that. It might be true or not. May be carried by a vector. Well, no, it's not carried by a vector. It's caused by a virus. That's the human immune deficiency virus. Carried by male Anopheles mosquitoes. Again, not true. It's the female Anopheles that transmits malaria. Measles can cause blindness. I'm not sure about that one, so I'm not going to make any choice there, but already that tells you B is wrong. Cholera is not an airborne infection. It can be caught by swimming in contaminated water. HIV is caused by a retrovirus. That's true. But again, because we already have one wrong here, we know that C can't be the answer, so let's not bother. And then here, causes reduction in the number of T lymphocytes. That's true. Malaria mainly kills children under 5. That's also true. No effective vaccine available. That's actually not true. There are vaccines for measles So that is a way to walk through very quickly. And here we know that our answer is A. Now let's do the last two questions. And I'm very glad that you guys have stuck through it. I know this is long, but again, in the exam, you would only have 1 hour to do this paper. So that's if you're writing in October, November. From next year, you will have 1 hour, 15 minutes to do this paper. So if you're writing May, June next year, congratulations, you have more time. But try to practice with 1 hour so that you're quick and you also improve your decision making. Which statements here describe myasthenia gravis? First of all, do you guys know what myasthenia gravis is? Since we're coming to the end, let's just take a couple of minutes to put something in the chat about that. If you don't know, just say, I don't know. No, just say, yes, I do. So yes or no, do you know what myasthenia gravis is? It's an autoimmune disease, that's correct. So an autoimmune disease basically means a disease whereby the immune system attacks the body of the person. Excuse me. And myasthenia gravis is an example of this. And so the answer here actually, the antibodies attack proteins within the body, that's true. Excuse me. I think there's some dust that just blew in here. T lymphocytes are involved in an inflammatory response. That's actually not true with myasthenia gravis. The immune system blocks receptors at the neuromuscular junction. Yes, that's true. If you want to learn about this, go and watch chapter 15. Not 15, chapter 11 of my videos. You will find them there. The immune system attacks the central nervous system. That's not true. So we are left with 1 and 3 as the correct answers. And so the answer here is A. The last question. I'm going to leave this one for you guys to just put in the chat. Just answer in the chat. Whoever is left. I know that some people have signed off already because this was quite long. Which row shows the cells that are able to divide continuously and are involved in monoclonal antibody production? Is it A, B, C, or D? Cancer cells, mouse B lymphocytes, hybridoma cells. Just going to give you guys a chance to answer that. Okay, so I see Y says C. It's the last question, so just give it your shot. Don't do eeny, meeny, miny, mo, okay? Think about it. What is involved in monoclonal antibody production? So I see A, all right. Come on, I see that there are quite a number of people. So just put in the chat what you think the answer should be. I'll give you like 20 seconds to just think of it quickly. Okay. So it seems maybe the others are a little bit shy because they're like, oh, everybody's going to judge me. Nobody's going to judge you for having the wrong answer. Think of these classes that we have as a community type class where you're able to express yourself and you can also learn if you don't know, okay? There's nothing wrong with not knowing. You learn the more if you don't know stuff and you tell people, you're like, so when you're doing monoclonal antibodies, you're actually going to use cancer cells. So that tells you that D can't be correct because D says no cancer cells. So we're looking at A, B, C, A, B, or C. You don't use the lymphocyte cells. The lymphocyte cells, the B lymphocyte plasma cells, make antibodies. So they are not involved actually in this production. Even though they make antibodies, they're not involved because what you do is that you link the hybridoma, the cells, the cancer cells, with what we call the hybridoma cells. Okay, and so in this case, that tells you that C would be our answer. All right, so you link cancer cells with the hybridoma cells. You don't link the cancer cells with the B lymphocyte cells. The hybridoma cells already have the lymphocyte producing cells and all of those things. But again, please watch chapter 11 on the channel. And that brings us to the end of this paper. I hope that you guys have found this helpful. If you found it helpful, can you just put a yes or a no? If you didn't, just put a no in the chat. It's fine. I won't take it personally. But it might just help me maybe refine how I do the live lessons next time. But if you found it helpful, just you can put a Y or an N if you don't want to spell it out. But I hope that it was helpful for you guys, and I hope that you've picked up some tips on how to study, like how to get through the multiple choice questions. Because believe it or not, the multiple choice questions are where students get the least marks most of the time. And in amongst all the biology, not the biology, all the science subjects for CIE, biology is one subject that students tend to fail the most. Students fail biology more than they fail chemistry or physics. And I'm not saying that to scare you. I'm saying that to tell you that the tips that you pick up from my videos or even from these live lessons are going to be very important, because they help you get through content very quickly, and they also help you quickly pick out options that are not possible so you can focus on the things that are possible. So I'm very glad that you guys find it helpful. Thank you so much for all the comments in the chat. I really appreciate it. I believe this would be available on YouTube. Well, I set it to be available. If it's not, then that would be really tragic, but I hope that it is. And I'm glad that you guys are finding this helpful. Please start preparing for the exams. Again, don't forget the tip I shared at the beginning. Watch a video on a specific channel, go into the past questions, and try to answer all the questions on that chapter. Okay, so if you go for chapter 1, for example, try to answer all the questions on chapter 1 and start asking questions in the comments so that people can respond to you and help you. All right, that's the whole point of this, to make sure that you get all the help that you need. Thank you all so much for sticking through it. We took 2 hours and almost 30 minutes, but I think we did a great job. So thank you everyone for being here, and I hope that you guys enjoy the rest of your day and have a great weekend. All right. Goodbye, everybody.